Hi guys, so today let's talk about chapter 18, the blood chapter. What is blood? What is blood made up of? Well, plasma and cells, right? So plasma makes up about 3 liters of your blood, and 2 liters of that is just cells. So that means you have about 5 liters of blood in your body, okay? And plasma is about 92% water. Okay, and cells? Well, we have red blood cells, right? Red blood cells that do gas exchange, oxygen and CO2. We have white blood cells that serve as our immune cells. And then we also have platelets that are basically fragments of cells. Okay? And their function is to aid in clotting, hemostasis, which we'll get to later. Alright, so we've talked about the constituents of blood, the general constituents of blood, what makes up blood, and down here we just have bloody function, or the function of blood, so be aware of that. Transport, oxygen, CO2, nutrients, and chemicals. Okay, and chemicals, that allows our blood to communicate, so it allows for communication and maintenance of homeostasis. So chemicals, like we just talked about in the last chapter, hormones are a type of chemicals that will communicate via the blood. Okay, the immune system with our white blood cells are neutrophils, eosinophils, basophils, monocytes. Okay, and then hemostasis. I keep saying hemostasis, but I mean homeostasis, pH of the blood. So normal pH is 7.35 to 7.45. Anywhere out of that range can be very devastating for the body. Ion concentration in the blood contributes to homeostasis. So ions are like calcium, sodium, potassium, and also body temperature. Okay. Body temperature is carried throughout the blood and contributes to homeostasis. Okay, all right. So we talked about what's in our blood, so let's look at that a little bit closer. So plasma, we said, is 3 liters. 92% of that is water. 7% is proteins like albumin. Antibodies are actually proteins. And fibrinogen, which is used for clotting. Albumin is the primary protein found in your plasma. Okay, it's made in the liver. So let's sort of draw some of these in here. So we have water, lots of water in the plasma. We have fibrinogen. We have albumin. Okay. We also have antibodies in the plasma. Alright, but that doesn't make up 100%, does it? So we have 1% left over. And that 1% is for ions like sodium or potassium, other nutrients like glucose. Okay. That are found in the plasma. So what about the cellular elements? Well, we have red blood cells. We have white blood cells. And then we have platelets, which are fragments of cells. Okay, so that's sort of a pictograph of your blood vessel and what is in it. So platelets, just a little side note here, platelets are, we said, fragments of cells, and they're formed from a bigger cell called megakaryocytes. Okay, megakaryocytes. They're really big cells. They have a hard time getting into the blood vessel itself, and so the pressure, actually, the blood pressure flowing through your blood vessel. So if this is a megakaryocyte, it's trying to squeeze through the joints or the connections between the epithelial cells, and pieces of it squeeze through, and the pressure, your blood pressure, the flow of the blood cleaves off pieces of that megakaryocyte, forming a platelet. Okay, so down here we have a centrifuge tube, or a test tube. And if we put that into a centrifuge, what happens is all the constituents of the blood, everything that is in the blood, gets separated out. So a centrifuge, if you sort of think of a washing machine tub, it spins, right? And whenever you look inside, all the clothes are pushed to the sides or the walls of the washing machine tub. So it works the same way. It's a carousel with the tubes placed in the carousel, and it spins really fast. And what happens is the most dense particles get pushed to the very bottom of the test tube. So 44% of those are red blood cells, and they're the most dense. So they end up being on the bottom. In the middle line, we have white blood cells and platelets. And then at the top, the lightest, the least dense, we have plasma, which makes up 55%. Okay? So the reason they do this is, one, to separate the components of blood, but measuring the amount of red blood cells can give you your what's called the hematocrit. Okay, the hematocrit, that will tell you your oxygen carrying capacity. So that means like how much oxygen you might have in the blood. So not enough red blood cells can also lead to things called anemia. All right. Okay, so this here is the family tree of blood cells. So up here at the top, we have the multipotent hematopoietic stem cell. Multipotent, that just means that it has multiple potential. Multiple potential means it can become many different things. So as it differentiates into different cells, we form different lines of cells. Okay, so then we have the myeloid stem cell and then the lymphoid stem cell. The myeloid stem cells become the megakaryocytes, the red blood cells, the erythrocytes, the basophils, neutrophils, eosinophils, and then even monocytes. Okay. The lymphoid line of cells become natural killer cells, and then T cells, or T lymphocytes, and B lymphocytes. Okay, so these start off in the bone marrow, differentiate into their different types of cells, and then eventually end up in our blood. So what you need to know from here is how they start out. So what that cell is called, the different lines here, and what we end up with. Okay. So this might look familiar from lab. It's a list of the different leukocytes, or white blood cells. And I just want to give you some facts about these. So the neutrophils, eosinophils, and basophils, those are your granulocytes, so all the phils. are granulocytes, meaning they have granules. Okay, so let's start off with those. Neutrophils are the most abundant. Their function is predominantly phagocytosis. So they like to eat things. Phagocytosis. Mainly of bacteria. Okay. Their granules have a chemical in them called pyrogen. And we'll talk about pyrogen later on, but be familiar with that, or be aware of pyrogen comes from the granules in neutrophils. Okay, eosinophils are cytotoxic. And their granules make things, well, their granules will pop out at you. They're very red, but or they stain very red. But they have chemicals called they have granules that are released in response to parasites and allergens. So you may, even though we don't normally have a lot of eosinophils found in our blood, you may see a rise in these when there's a parasitic infection, or there's the presence of an allergen. Okay, and then the basophil. The basophil, its function is predominantly inflammation. Okay, its granules produce a chemical called heparin. You may have heard this word before, heparin. It's an anticoagulant. It's an anticoagulant. I don't have room to write that, meaning it keeps things from clotting. It also makes histamine. Histamine. So you may have heard of antihistamine allergy drugs that help to dry you up. Well, this produces histamine, which is a vasodilator. Okay. Both of these granule chemicals will increase blood flow. Both of these will increase blood flow. Okay, let's talk about the other two. Lymphocytes, which are the littlest of the leukocytes. Their function is acquired immunity. Acquired immunity. And they form B cells and T cells, which we will talk about at length in the next chapter. Then monocytes, which are the biggest of the leukocytes, their role is phagocytosis. So they also like to eat things. Okay, they can eat 100 bacteria in their lifetime. So one of those monocytes can eat 100 bacteria in their lifetime. Okay, whenever they transfer from the blood into the tissue, they become macrophages. Become a macrophage, okay? Macrophages in the brain are actually called microglia. Alright, so that's a little extra information that you might not have gathered from lab that I want you to be aware of. Okay, so I may ask you some of these questions on your exam. Alright. moving right along, let's talk about hemostasis. So hemostasis, what is hemostasis? Well, that's the stopping of bleeding. So hemo, blood, and stasis means still. Okay, stopping of bleeding. So here we have a picture of a blood vessel, and our stimulus is going to be some sort of damage to that blood vessel and breaking of that blood vessel. So basically 3 things happen here. This will be 1 and this will be number 2. Okay, so I know they're numbered, but keep in mind that all of these happen simultaneously. Okay, so we get vasoconstriction or a myogenic response. Myogenic response refers to the smooth muscle in the blood vessels constricting. Okay, the smooth muscle constricts, and that's due to a chemical produced called thromboxane A2. That chemical may be familiar to you from our previous chapter. That's a eicosanoid. It's sort of a positive feedback mechanism here that we get vasoconstriction. Thromboxane A2 is produced, not really from the vasoconstriction, I'm sorry. It's actually from the stimulus itself, from the damaged blood vessel. That also causes platelets to become activated. Okay, and that in turn causes more thromboxane to be produced, causing more vasoconstriction. And in turn, those activated platelets form a platelet plug. Okay, this is not a clot. Okay, so make a note of that. It is not a clot. That platelet plug grows. They are irregular in shape. They are sticky, so they're sticking together. And then they produce more chemicals. Okay, that activate more platelets. So again, it's more of a positive feedback mechanism. Okay, so again, this is the platelet plug is not a clot yet. All right, so that's 2 things that have happened. So what's the third? Well, that's clot formation. All right, and this happens fast and furious. And the clot formation is termed procoagulation. All right, it causes a chemical cascade. So we get a chemical cascade happening. And if you notice in your book, when it talks about clot formation, it has all these different lines leading to clot formation. Well, I'm not going to ask you to know all those different chemicals and so forth. I've given you sort of the Cliff Notes version of it and what I'm going to ask you to be aware of. So just know that it causes a chemical cascade that leads to prothrombin activator. And that prothrombin activator takes prothrombin and activates that into, guess what? Thrombin. Once we have thrombin, then it can take fibrinogen, which is in the plasma, right? And is a protein. And form fibrin. And that fibrin joins with the platelet plug to form a clot. Okay? So we just did procoagulation. But realize that we also have anticoagulants in the blood. All right, so these are always found in the blood, the procoagulants, but we also have anticoagulants in the blood. Both of these proteins are found in the plasma at any time. It's whether or not one is found in more abundance. So it's a delicate balance between the two. All right, so if we have damage to the blood vessel, then we're going to have more procoagulants. All right, if we had too many of them, then when we didn't have a stimulus of a broken blood vessel, then we would be forming way too many clots, and that would not be good. It could lead to things like a heart attack or a stroke. So we also need to be aware of anticoagulants. And you may have heard of some of these, like aspirin. And aspirin is what's called a COX inhibitor. Okay, a COX inhibitor. So how that really works is, let me draw that over here. If this is the COX enzyme right here, it needs, guess, thromboxane to activate this enzyme. Okay? And what aspirin does is it comes in and blocks this mechanism. All right? And I guess this arrow should be going this way. Aspirin comes in and blocks this enzyme. And if we don't have thromboxane, then we don't get any of this. We don't get clot formation. We don't form this platelet plug, right? What about Coumadin? What does Coumadin do? Well, Coumadin is also known as warfarin. Okay, it's also known as warfarin, and it decreases vitamin K. Decreases vitamin K. You're like, I have not seen any vitamin K on here. Well, let me show you where it's at. Vitamin K is needed for the liver to make this protein, prothrombin. And if we don't have enough vitamin K, then we can't make prothrombin. And if we can't make prothrombin, then we never get thrombin. Okay? What about citrate? Citrate is also an anticoagulant. They often give you citrate whenever you donate plasma. Well, citrate deals with calcium, so it keeps calcium from being absorbed. Calcium is needed in nearly every single one of these chemical cascade pathways. Okay, so if we don't have calcium, then we don't get prothrombin activator. All right, so be aware of all of these proteins that I have listed here, and then also the anticoagulants and what we would not make if we took, say, aspirin or Coumadin or citrate. Okay, let's keep moving on. All right, so this picture shows erythropoiesis. Erythropoiesis, what is that? Erythropoiesis is the production of red blood cells. Okay, red blood cells, they're made in the bone marrow. So we start off with the stem cell, like we showed in the family tree. it becomes a proerythroblast. A proerythroblast. I'll abbreviate that. Then it becomes an early erythroblast. Then finally an erythroblast. And then a normoblast. These crazy names. And once it becomes a normoblast, it stays here in termed a reticulocyte for about 3 days. So here's our reticulocyte. So let me say that again. So as a normoblast, once it becomes a normoblast, it turns into a reticulocyte. It stays in the bone marrow for about 3 days, and then it goes into circulation, into the blood. And here's where it loses its nucleus. Okay, so when it's still in the bone marrow, it has a nucleus. Once it is in the blood, it loses its nucleus. Okay, if it had a nucleus, it would want to hold on to oxygen, and that would not be good as a red blood cell. So now here is our actual erythrocyte, or red blood cell. Red blood cells, they last how long? So they last about 120 days. So they live anywhere from 100 to 120 days. But eventually, they get old. And so what happens as they age? Well, things take care of it, like the spleen, the liver, and even the bone. And macrophages of the spleen, liver, and bone do this. And this is the sign for macrophage, an M-O with a line in it. That means macrophage. Remember, those are monocytes when they're still in blood circulation, and then when they're in the tissue, they become macrophages. So what happens? So if we have here a macrophage in the spleen, and here's our old red blood cell, it gets broken down, and it takes the hemoglobin, and breaks that further down into globulin and the pigment heme, which is that red pigment. That globin gets broken down into amino acids, and the heme pigment gets turned into iron, or broken down into iron, and also bilirubin. Okay. So be aware of the cells that do this, that break down the old red blood cells. So here I have the chemical structure of hemoglobin. Hemoglobin is abbreviated as HB. Okay. Next I want to talk about oxygen saturation. So one red blood cell can contain millions of hemoglobin. All right? And that hemoglobin is where oxygen is found. So let's draw some hemoglobin. I'm going to draw them like little flowers. With the red heme pigment in the middle. Okay. Hemoglobin's made up of the protein globin and bound to that red heme pigment. So if we take a look at one of these guys, they're made up of 2 beta globulin chains. So there's one beta globulin chain. Here's another one. and I'll do the other ones in green. And here's the alpha globulin chain. And each one has the red heme pigment in the middle. Okay, so that's a beta, that's an alpha globin chain. All right, so hemoglobin, we know contains 4 globin chains, and we know which ones those are. Here's the chemical structure of hemoglobin, and this iron right here is what actually binds the oxygen. Okay, when oxygen binds to iron, the iron of the heme group, it becomes oxyhemoglobin. So I'll write that for you. Oxyhemoglobin. When oxygen detaches from that iron in the heme group, it becomes deoxyhemoglobin. Okay, deoxyhemoglobin. Be aware of those definitions. All right, so I'm not going to ask you to know that chemical structure. It was just for understanding purposes. All right, let's keep moving on. Now I want to talk about ABO blood typing. And I know your picture looks a little bit different than this. Let me erase these real quick. We'll talk about this. Okay. So we need a good definition of antigen and antibody. So what is an antigen? Well, it's anything that can cause an immune response. Okay. And an antibody, what's an antibody? Well, it's a plasma protein, we know. And it looks something like this. And it has long chains and short chains. But at the end of the short chains, it has this variable region, VR for variable region. And those variable regions are super diverse. They're specific for every single type of antigen, and they're made by what's called gene shuffling. So it allows those variable regions to be very diverse, okay? And that antibody matches to a specific antigen. Okay, so now let's talk about blood typing. So we know, I'm going to start off with just the ABO and leave off the negative part for now. So we know we have type A, type B, type AB, and type O blood types, right? So if this is our red blood cell, and in type A blood, type A blood has what type of antigen? Okay, so antigens are found on cells, right? And antibodies are found in the plasma. So type A blood would have type A antigen. What about type B? Well, type B antigen. And AB would have type A and type B antigens. Type O, well, no antigens. That doesn't mean to say it has no antigens whatsoever. It just means it has no AB antigens. Okay? So now what about antibodies? Well, we have two different types of antibodies. We have type A antibody and type B. And what that means is anti-A antibody or anti-B antibody. That means it's an anti-A to the to the A antigen. So with type A blood, it would have what type of antibody? It would have a anti-B antibody, because if it had an anti-A antibody, it would attack its own red blood cell. With type B blood, it would have an anti-A antibody. What about with AB blood? No anti-A or anti-B, because then it would attack its own red blood cell. What about with type O? Well, we have no antigen, so we have anti-A and anti-B antibodies. Okay. So I want you to ask yourself these questions and be able to answer those. All right, the antibodies are termed IgMs. These are immunoglobulins, they're proteins, and I remember this by M means massive, okay? That they're massive and they cannot pass the placenta. That's going to be important for the next thing. Okay. Now with the negative factor, that's the Rh antigen. We have antibodies to those. So if this is an Rh antibody, or they call them anti-Ds. I don't know why they call them anti-Ds, but they do. These are IgG immunoglobulins, and they can pass the placenta. They are small, they're tiny. Okay, so let's do an example here with our type A blood. So let's say that we are type A positive. So that would mean not only do we have an A antigen, but we also have an Rh antigen. Do we have an Rh antibody? Well, as far as we know, no, we don't. Okay. Now let's talk about if we have a baby. Okay, so here's our pregnant mom. She looks miserable. All right. And she has a little baby inside of her uterus. And this is the placenta here. Okay, and the placenta is part mom and part baby. And mom and baby's blood never mix until birth. So where chances are that whenever the placenta rips from the side of the uterus, there will be some mixing of blood. Not always, but there's a high probability of this. Okay, so let's take a look at mom's blood type. So let's say that mom is, let's say she is A negative. Okay, so what does she have? She has an A antigen, right? Does she have any Rh antigens? No. So what antibodies does she have? Right, a B antibody. Okay. Let's make her, I changed my mind, let's make her AB negative so that she has, oh, sorry. Let's make her O negative so that she has no antigens and both A and B antibodies. Okay, but no Rh antibodies right now. Okay, now let's look at baby's blood. Okay, let's say baby is A positive. That means baby has an A antigen and a B antibody, but it also has a Rh antigen, right? Okay, so everything's okay right now because we don't have any blood mixing, right? Okay. All right, so baby one's okay. So let's say that mom gives birth, there is some ripping of the placenta, and we do get some blood mixing. All right? And mom is exposed to this Rh antigen. Now mom is going to make an Rh antibody. All right, well, baby's not in there right now, so it won't attack this. But let's say mom has another baby. And let's say this baby is also A positive. So its blood would look like this, with an A antigen and an Rh, right, and a B antibody. So mom is making Rh antibodies. Now mom is making antibodies against this antigen. And the reason this causes a problem is because Rh is an IgG, which we just learned can cross the placenta. And that causes mom's body to attack baby number 2 as something that is not supposed to be there, and it can actually kill the baby. So what has to happen is that mom has to get a drug called Rogan. And what that does is suppresses mom's immune system to stop making these Rh antibodies. All right. So practice your ABO blood types and RHs by drawing some blood vessels with blood cells in it, and give yourself some scenarios and see if you can answer the questions, because they will be on the exam. All right, there's one more thing I want to talk about and we'll be done. And that's erythropoietin. Erythropoietin is a hormone, or EPO. It's made in the kidneys, in the cortex. So this is the cortex of the kidney, the lower portion of the cortex of the kidney. And over 1 liter of blood per minute goes through the kidney. And those special cells in the lower cortex of the kidney sense oxygen, O2, in the blood that's going through the kidney. Okay? And let's say that they detect low O2, low oxygen levels. The kidney produces this hormone, erythropoietin, and if it senses low oxygen, it will increase secretion of EPO, erythropoietin. This EPO, since it's a hormone, gets dumped into the blood, it circulates, it targets the red bone marrow, and stimulates erythropoiesis, which we just learned is the production of red blood cells. Remember, these red blood cells are carrying oxygen, which was our initial problem, right? And when these red blood cells return back to the kidney, carrying the increase in oxygen, it will then decrease the secretion in EPO. So it's a negative feedback mechanism. Okay? Renal patients, people that have kidney disease, will be given EPO to stimulate red blood cell production and increase their red blood cell mass. Because if their kidneys are damaged, then they're not being able to sense O2 levels. Athletes will sometimes use erythropoietin in what's called blood doping. So if they're taking extra EPO, they're increasing their red blood cells and in turn increasing their oxygen carrying capacity to gain an edge in whatever sport they're competing in. Okay? Alright, so that concludes our blood discussion. I hope this helps. So happy studying.